Protein synthesis and how cells make life happen. What is protein synthesis? Protein synthesis is the process where cells create protein. It happens in two main steps, transcription and translation. Protein controls nearly everything in your body, from muscle movements to the enzymes that help you digest your food. Transcription. Where does this happen? It happens in the nucleus of the cell, and the goal is to copy DNA instructions for a gene into a form that can leave the nucleus. Initiation. The enzyme RNA polymerase binds to a specific region of the DNA called the promoters just before a gene starts. It unzips the DNA's double helix in that region. Then we have the elongation stage. RNA polymerase reads that template strand of DNA. Only one strand is used, and it builds a strand of messenger RNA, which we call mRNA, by matching complementary RNA bases. We have A on a DNA strand and U on an RNA strand. Then we have T to A, C to G, and G to C. Here is a codon wheel. It is a tool used to figure out amino acids and what it is coded by a set of 3 RNA bases called a codon. Then lastly, we have the termination. When RNA polymerase reaches a termination sequence, it stops transcription. The new mRNA strands detach and leave the nucleus through a nuclear pore. Things to note is that the DNA is a permanent genetic blueprint, mRNA is a temporary copy of it, and RNA polymerase is the enzyme that builds the mRNA. This is a diagram of transcription. It shows the DNA strands is used to make mRNA. First, the enzymes of RNA polymerase attaches to the DNA strands that starts reading one strand. As it moves along, it builds a strand of mRNA using RNA bases. The mRNA strands then carries the genetic message out of the nucleus. Translation. Where does this happen? It happens in the cytoplasm, specifically the ribosome, and the goal is that the mRNA code to assemble a chain of amino acids which fold into a protein. 1. We have initiation. The mRNA attaches to a ribosome made of RNA and proteins. The ribosome reads that start codon and usually is AUG, which is methionine. Then we have the elongation. The ribosome reads the mRNA codon, which are a set of 3 bases at a time, and each codon corresponds to a specific amino acid. tRNA brings the correct amino acid. Each tRNA has an anticodon and amino acid attached to it. We have an example. mRNA is a codon for AUG, and tRNA is an anticodon for UAC. An amino acid is methionine. The ribosome links to the amino acid together with a peptide bond. And lastly, we have the termination. Translation ends when the ribosome reaches a stop codon, UAA, UAG, or UGA. The completed polypeptide chain detaches and begins to fold into a functional shape. Things to note is that codon's a 3-base mRNA word that codes for an amino acid. Anticodon's a complementary 3-base sequence, and ribosome is a protein-building machine, while tRNA brings the amino acid to the ribosome. This is a diagram of translation. It shows how mRNA is made during a transcription is used to build the protein now. The mRNA goes to the ribosome where it leaves its codon, and each codon matches a tRNA carrying an amino acid. The ribosome links the amino acid together to form a protein chain. This happens in the cytoplasm. Protein structure. Proteins are polymers, and their building blocks are amino acids. There are 20 different amino acids in living things. Each amino acid has the same basic structure: a central carbon atom, an amino group, a carboxyl group, and a hydrogen atom, and an RNA side chain. This part is different from each amino acid. Why is protein synthesis essential? Every structure and function in the body is built or managed by protein. Proteins include enzymes that speed up chemical reactions, hormones with chemical messengers, antibodies with immune defense, muscle fibers, skin, hair, nails, and even insulin is a protein hormone. If the DNA instructions for insulin are wrong, your body can't regulate body sugar properly, leading to diabetes. In conclusion, protein synthesis is how your body takes instructions written in your DNA and turns them into proteins that do everything from digesting food to fighting infections, in fast, complex, and life-saving processes that happen millions of times per second inside your cells. Thank you.